where we all meet together and have a nice discussion on basics of CompCamp and MedCamp. This is not designed to be an introduction because I think it would be a little bit too boring to everybody. So I tried my best to cover areas or papers, publications, case studies that are not so prominent to also open up some awareness in regards to certain aspects. And therefore, I would like to highlight applicability of bioisosteres in drug discovery in general. There are some misunderstandings around bioisosteres and functional groups in themselves that I would like to clear up here to also make you more successful in your job and also to have some a little bit more of fun on my side. Quite important for me as well to have some form of expectation management when it comes down to compound design at the end of the day, and to provide you with valuable insights. When I started the whole concepting of this workshop, I wanted to cover a little bit more groups, but it became evident that it would blow up the whole workshop, and to keep it in a sophisticated time frame, I limited myself to only 2 groups today. And when we talk about those groups, I would also like to conceptualize versatility of structure-based approaches when we talk about replacing functional groups. So to dive into the material, that's my laser pointer. Just to have everybody on the same page, when we talk about functional groups in medicinal chemistry, we are talking about a specific arrangement of atoms that lead to their chemical reactivity, their interaction potentials within the target binding site, which also contribute incrementally into the physical, chemical, and pharmacokinetic properties. Functional groups, I mean, you know them just as well as me, or even better or worse, depending on the case, can be carboxylic acids, aliphatic side chains, sulfonamides, Michael acceptors. So the whole toolbox of functional groups is still expanding. It's not like we are somewhere on a stagnation here, but we see reports here and there that people still explore new functionalities, which is nice. And therefore, it's always nice to be up to date when it comes down to this. Functional groups are the most important part of a molecule, which is a little bit dumb to say because honestly, every molecule just consists of atoms, and therefore it's just part of the molecule. It's just kind of like smaller segments of the molecule. But nevertheless, what makes them so important, which is also reflected by the Paul, they can form interactions within a target binding site, right? So they can form H bonds, they can form hydrophilic, lipophilic interactions, and therefore are the driving force for a ligand to actually bind the target structure and to modify its activity. Which is actually the thing that you and me are doing on a daily basis, to think about how you can actually improve binding affinity of a ligand by introducing additional functional groups to form more interactions in the binding site that are also of high quality. Which is also even more important if you work within the pharmaceutical industry, is to think about metabolism bioavailability. In academia, it's also part of the whole discussion, but people in academia can live within the carboxylic acid, whereas this is something that is a little bit more avoided in pharmaceutical industry because the metabolism there is quite high, and therefore the half-time shortens quite a lot. There are also unwanted groups, unwanted functional groups, which is basically something that is present in functional building blocks because they are highly reactive and therefore can be coupled to other building blocks to create compounds. So this is something you would not use, obviously, in medchem-like compounds because they would also interact with unwanted targets and therefore not something you would like to, because the toxicity is quite unaccessible here. I am a very lazy person, to be honest, when it comes down to collecting data. Therefore, it's always nice to work with nice literature, and therefore I picked up this one by Peter Erdl, previously Novartis, who looked into the most frequent functional groups used in bioactive molecules. And if we dive into that, we get this list of now 40 most frequent functional groups. And I don't know, for me, it was a little bit surprising. I didn't expect it to be that high. The most prominent one is the amide, followed by ether, amines, and so on and so forth. So therefore, I thought, why not actually think about the whole functional groups in the context of our software and our tools, I keep it as salesy, not as salesy as possible, but I also looked into our fast score functionality and especially the METCAM set with 120,000 fragments and compared the occurrence of functional groups within this data set just to get a feeling, because honestly, we haven't checked it beforehand. And this is what emerged here. So in blue, we have the percentage of the Apple assessed set, and in green, we have the fast 120,000 library. And I'm actually quite surprised that his observation is also nicely reflected in our Pascal library. So you can also see how the amide is the most prominent one, and then it almost kind of like adapts nicely. There are some exceptions like the double bonded compounds here, but all in all, the bioactive compounds or the distribution of functional groups around them, it's also reflected nicely in our FASCO library. So you might have guessed it. The first thing I will talk about is actually the amide, which is the most prominent functional group. Some basics, the pure amide is actually planar. So 120 degrees for all heavy atoms. It's in one plane. It's quite flat. And in the context of peptide chemistry, it's also referred as a peptide bond. Same thing, but just in the context of amino acids. There are a lot of reasons why the amide in itself is the most prominent functional group, namely because it's synthetically quite accessible. So the most prominent reactions are very, very straightforward. You have an amide, you have a carboxylic acid, both are the most frequent functionalities inside building blocks. And what you do, you use a catalyst, couple them, voila, you end up with an amide. There are also a lot of other functionalities, reactions that can be used for the synthesis of amides, just to name a few. What's quite important for me as well, multi-component reactions. So the UGI reaction uses 4 building blocks, and if you couple them together, you end up with 2 amide functionalities, and the decorations of the building blocks in between them and on them, which is quite nicely if you want to create larger libraries. Given the fact that you multiply in a combinatorial perspective, 1 times 2 times 3 times 4. So this really blows up numbers of combinatorial compound libraries. Now to dive a little bit more into the 3D world, for me, it's quite important to also expand a little bit on the case studies here, not just showing you something boring, but also everything that I will talk about. I try to supplement it with real PDB data, real examples, data from bioactivity data. And in those 2 images, I just wanted to highlight how an amide can actually interact with the target structure. Quite important, we have here an aromatic ring system, pyridine, and then an amide. Both are in the same plane due to the electron distribution. You can see nicely how the amide can engage in H-bond interactions. The oxygen itself is eligible to 2 oxygen interactions, in this case with the tyrosines here, and the amide only 1 H, therefore only 1 interaction, now here with an aspartate. Really important, the natural occurrence of amides is a trans configuration. So basically, we're talking about this carbon and this carbon. Both are not on the same side, but on the different sides. So this is how a natural amide looks like, trans configuration. In the case of Valdaprevir, developed by Böhringer Ingelheim for the treatment of hepatitis C infections, again, we can see how nicely the oxygens and the nitrogens can interact within the binding site. Geometry is a quite important factor for the interactions of the oxygen, as well as for amine, for the amine here, for the nitrogen, sorry. It's pretty straightforward, so 90 degrees for the perfect arrangement, whereas for the oxygen, it can differ a little bit, but there are only certain combinations that work nicely. And I would like to emphasize here once again, in our software Caesar, you can actually distinguish the quality of H-bond interactions of the interactions due to the color. In dark green are high quality H-bond interactions, whereas in light green not so the perfect geometry. Cis amides do exist. This is something we should not neglect and we should be aware of. This is an example 3-NTP. This is a cis configuration. What you can see here, this and this carbon atom are on the same side, therefore the cis configuration. It's quite rare to occur naturally, to be honest. Yet in some microcycles, especially, this can be observed. So ultimately, if you look also into the histogram distribution of amide bonds, you can actually see that the natural trans configuration is the most prominent one. Yet there exists the cis configuration, the PDB files and the PDB formats, and it's also this second peak here and here. Okay. So just because the amide is the most frequent one doesn't mean that it's perfect. There are some downsides to that. Amides are cleaved, so therefore the metabolic stability is limited. And this is where biosteric replacements can be useful to make to prolong the half-life of a drug. The second thing is that even though it is actually designed to interact within the binding site due to its H-bond donors and acceptors, it also reduces the permeability of compounds containing an amide. This is something that can be addressed if you introduce intramolecular H bonds into your compound, which will be presented on the next slide. Especially if you have if you have a lot of amides within a compound, like in peptides, this has potential to trigger immune response due to the formation of haptens. So basically, the amide attaches itself to another protein structure within the human body, which is then recognized as an yeah, how do you say it in English? It's something that's not supposed to be inside the body and should be degraded, which can trigger an immune response. The simplicity of the amide, it's only 3 heavy atoms, brings a lot of problems in regards to drug design. So therefore, it's quite difficult to find something of the same size, of the same atom arrangement that satisfies the interaction within the binding site if the amide is important. Therefore, it's quite difficult to really substitute or replace an amide easily if it's a linker. This is something that will be addressed in the subsequent slides, where we talk about geometric scaffold hopping or morphing to come up with decent ideas how to replace an amide. And last but not least, the H-bond donors and acceptors also make it low compatibility with lipophilic targets due to its hydrophilicity. In this slide, I wanted to highlight some perspectives on intramolecular H bonds. This is something that amides are quite well known for, I would say. So basically, by forming intramolecular H bonds within other heteroatoms within the binding site, within the ligand, pseudo-ring systems result. Those things stabilize active conformations of this of a ligand and are very hard to break through interactions. So basically, once you introduce an H bond within a molecule, you actually fix this orientation, and therefore it's really hard to make it flexible subsequently when it's needed for a binding site. But it's quite nice to make a compound also rigid, reduce lipophilicity, make it more membrane penetratable, and so on and so forth. What we should be aware, though, that proximal, which means close neighborhood interactions, are very strong. So to break those kind of things, close to impossible. Distant ones, though, are a little bit different. So if you look at the crystal structure of the previously mentioned Valda-Previr, we can actually see that in the CSD file, we can see a nice intermolecular H-bond interaction occurring here with the carboxylic acid and this amide thiazole profile here. Yet this is not something that we observe in the PDB file. So if we look at this PDB, we can actually see how everything adapts basically because the potential, if this one interacts with the ligand, with the target binding site, and the carboxylic acid interacts with the binding site, we have way more energy that we can get out of that. And therefore things try to, open up to engage in more interactions, which can we see here in this 2D interaction diagram of the PDB file. So now to dive into the more interesting part, what is a bioisostere? To be honest, at this point, nobody knows because it's used in a lot of contexts and for a lot of things. Ultimately, we are talking about a replacement of something, of a functionality within a molecule that has somewhat similar or improved biological activity or physicochemical properties. This is more of a concept, right? So basically, this is something that just occurs. You see, okay, this compound has not the best metabolism, not the best binding affinity. What could we change to improve this? So you look up cases, okay, how can I exchange this amide? How can I change this, I don't know, triazole? You just try things out, and whatever matches becomes a bioisoster for you. Which leads us to the whole scenario, whatever works, works. So basically, whatever works is a bioisoster by the end of the day. And if we take a look into this example here, we can see we have this pyridine with an amide and an aromatic amine, which can be basically substituted for a second amide here and also the cyclopropyl here. Ultimately, the bond length here and here are the same. This conformation is quite fixed, and the resulting affinity of both compounds is more or less the same. Yet, if you would look at that, what's the bioisoster here? Ultimately, we got rid of the whole aromatic system here, which means it was not necessarily needed for proper interactions. And the only thing that remained from here is the bond length, the geometry, which is quite important, and this H bond acceptor, here the nitrogen and here the oxygen. The good thing about bioisosteric replacement is that the number of atoms that are replaced is not limited. So you can go from 3 atoms to 5 to 10 ultimately, as long as it just works, you know. And bioisosteric replacement allows you to work with a lot of freedom. Let's put it like this. You can change the number of H bond acceptors and donors as long as tolerated. You can also kind of like interchange between sp2 hybridization, sp3 hybridization, so from aromatic to aliphatic and vice versa, as much as you want, as long as it works. And even formal change is also tolerated in some cases here and there. The only thing that remains is you just want to maintain potency and selectivity, the physicochemical properties, and the ADME parameters. So what are the reasons to introduce a bioisoster? To be fair, those parameters on the right are fluent, so the limits, the borders are fluent here. You can pick one and place it somewhat into another as well. So don't nail me down on the categorization here. So ultimately, would like to improve molecular parameters like binding affinity, interactions with the target, H bond donors, rotatable bonds as well, which is also kind of like a fluent bridge to also Lipinski's rule of 5 together with the next one, which is namely the physicochemical properties, which can be lipophilicity, solubility, permeability, and stuff like that. What also is quite important is the pharmacological behavior and ADME properties. So ultimately, when you start to introduce bioisosteres, you are actually quite progressed. So you have already some insights into the structure-activity relationships, where you really try to fine-tune especially those parameters by introducing bioisosteres. So basically, half-life is quite important, permeability again, metabolic stability. Also not uninteresting is the toxicity mitigation, so basically reducing toxicity. There are a lot of things that should be considered, obviously. Mutagenicity, quite rare, but can occur. But the most prominent one is actually the CYP inhibition, because some groups are heavily metabolized, some others are not. And what is the most important factor for me ultimately nowadays is actually to introduce novel IP. Especially if you look into patents, patents do work quite a lot with very fixed structures because they are aware of what works and what does not work. So if you're looking at something, a patent, you would like to burst it, bioisosteres can be actually quite nicely used to circumvent certain limitations when it comes down to patented structures. So quite important as well that bioisosteres are always a nice way to increasing the coverage of the chemical space that we know. So ultimately, bioisosteres are there to enhance all aspects of drug performance. So if we think about the selection of a bioisostere for a molecule that we are working with, we should keep some things in mind. It's not so easy to actually maintain interactions for a lot of reasons while also maintaining molecular weight of a compound. So on the right, we have a selection, only a selection, not everything, only a selection of carboxylic acid, and that's bioisosteres. They're quite commonly used. What they all have in common, except for this one, is in the physiological pH of 7.4. All are deprotonated, so therefore charged, which is basically just a promiscuity aspect of why they are considered as bioisosteres by the end of the day. It not goes also for the sulfonamide, can be also neutrally charged, but you get the gist. So ultimately, the promiscuity of those compounds contributes why they are considered as bioisosteres, and they're kind of like interchangeably. So if you have a phosphate, people also try to work with carboxylic acids, and this is kind of like, I call it a true compound functionality, because it's never used in drugs, but if you have an academic group and they just kind of like try to explore the target, every now and then you see sulfonamides used, but yeah, not for medchems like. So ultimately, in theory, on paper, all of them are equal, but there's a huge but, namely the geometry here. So ultimately, the carboxylic acid is planar, as we know, but if it really should satisfy a binding site, things become difficult. The tetrazool here is also quite planar, which is also contributing to the fact that it's the most prominent and the most beloved carboxylic acid bioisostere. But if you take a look at sulfonic acid, sulfonamides, and the phosphates, you see that the geometry changes. We are not flat any longer. This is an tetraidic structure here. And therefore, it highly affects the interactions with the binding site, which you should keep in mind because it affects a lot of things, which we will talk about later. So ultimately, quite important, 2D replacement can work, but for me, it's one dimension too short. If you have the luxury of having somewhat an access to a 3D structure-based approach, always rely on the 3D approach because this works on paper, but can fail quite nicely, which we will see also in the subsequent slides. Ultimately, if you are thinking about bioisosteres, you also should keep in mind the synthetic feasibility and the availability of building blocks. Amides in this case, quite cheap. You have a lot of amides, you have a lot of carboxylic acids. But if you start looking into tetrazoles or even sulfonamides, things can get quite expensive pretty fast. So again, something you should be aware of when designing compounds and deciding on your next steps. I won't go through every single one, don't be scared. This is something that we have worked on the past few months. I've collected quite a lot of amide bioisosteres. You can also access those slides subsequently when they're available as download. Ultimately, there are a lot of things you can work with. What's quite important here, this is the amide, and I took a little bit of effort and also calculated parameters just for the increment of the bioisostere itself to make it somewhat comparable to the amide, to give you a little bit more help if you want to decide what bioisostere for the amide you would like to work with. In this case, it was designed for peptides, but if you just ignore the R group here, it works quite nicely for you as well to make decisions based on which one of them you would like to use in your compounds as well. There's also a second slide to that. What I would like to highlight here as well, what works for an amino acid does not necessarily work for a proline, because the proline is a little bit special. It's a tertiary amide, having two things, less stereochemistry in this regard, less flexibility. So I also took some effort and collected some proline bioisosteres. If you fancy that, again, 1 and 2 slides to work with. Which is quite interesting, just to have everybody on the same page. If it's pretty close to the original value of the starting point, everything is gray. But if there are changes lower to the number, which makes it higher or lower, it is highlighted in blue and red, respectively. Okay, why do I talk about that? In December, we released the dipeptide ASCO library. Basically, it's two libraries, one starting with the N terminus and one starting with the C terminus, where we use proteinogenic amino acids and bioisosteres to actually create a dipeptide set, 660,000 conformations, not conformations, sorry, entries, 10 conformations each. So if you work with peptides, quite efficient, quite helpful for you guys. So please go ahead and download it if you want to. Okay, enough of the advertisement. Now into the case studies. This is something I picked up as a starting point to discuss larger, the whole picture, right? This is something that was published by Luot Fan et al. From Bristol Myers Squibb in January 2023, where they had this beautiful compound in front of them and thought, okay, how can we improve this one? We have two amides here, the hinge binding motif here, and what they observed in the crystal structure is that both amide actually interacted with a conserved water molecule here. So they thought, okay, how can we approach this? How we can increase the permeability and actually get something that is also blood-brain barrier penetrant. And they merged. So basically, they merged this amide scaffold into this heterocycle here. And now this heterocycle formed an intramolecular H bond. This got rid of the water molecule. So basically, this interaction mimics this water molecule interaction to some degree, and also covers this hydrophilic part of the molecule, which increased the blood-brain barrier penetration quite a lot. We also added an morpholine ring layer for solubility purposes and ended up with a compound that was even more potent than the initial one in the picomolar range now. Both stories published by Myers Squibb, this one in January 2023 and this one in March 2023. For the next miracle, I need a little bit of more prologue. So this is an interleukin antagonist, compound 1, which made quite some transformations over the years. This was the first one that was actually used to crystallize interleukin 17A back in 2016. And follow up by Novartis, they published two studies. This one in September 2024, and 3 months later in December 2024. So on December 23rd, so one day before Christmas even, they also published this one, compound 6, which I'd like to talk about. So the original compound, we can see pretty nice interactions of the amides, again, transconfiguration, planar, which forms two H bonds with the lysine 120, the backbone of it, from the dimer. And if we now take a look how compound 1 compares to the others, we can actually see that in this case, we have this pyrazole on the left, and then this longing tail to the right. What Novartis did on their side, they actually replaced, got rid of this tail here. You see it's kind of like shortened, and then added a little bit more of fancy stuff on the left side to project into another sub-pocket. And what we can see here is actually that the amide here is now replaced by this heterocycle that forms an H-bond interaction to the oxygen of the backbone here. Additionally, we can also, if I turn it around, we can see nicely that this cyclohexyl group here that is acting as a bioisostere of the phenyl group. And if we take a look into the other compound series, other compound series, there you go. Again, we can see quite nicely how the thiazole is placed here, mimicking the carbon atom of the amide from the compound 1. And if we now put it in the whole perspective, you can see how nicely there are actually two different scaffolds mimicking the original amide here. Those are the PDB structures, the heterocycle, the diazole amide, and also a derivative with here with an extended ring system on this part. What's quite important in this case is that this compound was reported as a quite prominent example for chalcogen interactions. So basically sigma hole interactions between the sulfur and the oxygen atom here. There are some more examples on certain conformer stabilizations. Ultimately, what happens here is that the orientation of the sulfur is fixed here, so the sulfur can no longer flip over. And due to this internal energy gain of this intramolecular interaction, this particular conformation is stabilized, increasing the potency of the compound. For my personal assessment, I would also say that ultimately it's actually the nice interaction pattern that is still maintained within the binding site with the backbone here. I wouldn't say necessarily that this is the main driving factor for the success of the story. Yet we should be aware that sigma hole interactions are existing and are actually a nice thing to get a more rigid active conformation of a compound. To make this long story short, we can nicely see here how the amide was actually replaced quite a few times to end up with compounds with better affinity and also better physical, chemical, and pharmacokinetic properties. Okay, again, a little bit of background information, geometry of amides. What we see nicely here, if we have an aromatic ring system followed up by an amide, they're both in one plane. Yet, if another one comes, we have a problem because we get clashes. So this is something that this compound does not like, energetically highly unfavorable. So what the amide does, or the whole system does to some degree, so they allow some smaller rotations of each one to actually maintain the good torsion quality of a compound. So amides are not always in one plane when it comes down to aromatic ring systems, especially if it leads to clashes. Things change, and things change quite a lot when we actually methylate an amide, which is a common strategy for for adjusting the metabolism of a compound. Because if we put a methyl group on this amide, it's way more difficult to cleave, and therefore the half-time of compounds increases significantly. What happens if we introduce an methyl group into the amide? We can see quite nicely that we actually get cis conformation. So the naturally occurring methylated amide conformation is a cis conformation, which you can also see in those both cases of the PDBs. Cis conformation and cis conformation. Obviously, it becomes even more complicated if we have two ring systems to the whole thing, because we now have not only the previously mentioned clash, but another second one. So therefore, we can see in almost all PDB structures how things completely twist by 90 degrees. In this case, linalolamide, we can see nicely how both are completely different planes, both turned by 90 degrees. So this in-plane, out-of-plane distortion results in no clashes for the compound, which ultimately results in more activity and NG favorable conformations. So now into the amide bioisosteres. This is now highlighting a very, very important geometrical aspect of bioisosteres. In front of us, we have two BDBs, again, and kinase inhibitor, as far as I can recall. And what's quite important here, we see nicely how the amide in this subsequent and this distal subpocket forms a pretty nice interaction pattern here now with an aspartate and with a water molecule and the lysine here. And now we have this triazole as an bioisostere, and it forms also quite nice interactions, this time without the aspartic acid though. So if I show you this graphic, you would say, yeah, well, looks nicely, right? There is nothing complicated. There is interactions. What's the point? The point is the geometry. If you now check for the planes of the amide and the triazole, we can see that they're both turned by 90 degrees. In the amide case, we can see nicely how it actually flips up outside the plane of the triazole. And if you now have this, very reduced visualization, we see that they're both in absolutely no plane. And this is quite important because if you would design something in 2D, you would say, well, it would bind the same way. But in reality, things change. Why is this important? If you extend the whole idea, you would say, okay, I have this compound, I have this compound. Obviously, the H bond acceptor here and here will have some interactions, but we have this ethyl group here. So introducing this ethyl group into this triazole, there is no data published on that, no data published on that, but we should keep in mind that the whole triazole changes. So to really host this ethyl group, it would need to flip out the plane. Both are aromatic systems, so therefore they would like to keep at least in somewhat in the same plane, but this would lead to clashes. So therefore, it's really hard to assess if the thing, the ethyl group that worked in this compound will also work in the second compound of the bioisostere. This is something that you should always be aware of when you just introduce something into a compound, because if there is something attached to that, you should keep in mind an bioisoster will always change the geometry. So how do we put this into motion? I've just picked a random PDB with an amide, namely this one, and thought, okay, how can we be more efficient? Okay, not only looking at the amides, but really cover the whole system of the compound, the whole idea, the whole concept. And for this, I used, again, some data from Peter Erdl. He published a nice review, a nice collection of bioactive ring systems. And this is something that we have also processed into FastGrow library, not FastGrow, Recall library, which we can use for 3D rescaffolding. So I took this compound into CSAR and used Recall in the inspirator mode. And what I did, I tried to keep the amine here and cleaved anything from here to here to check, okay, what 3D geometric replacements are out there that satisfy this binding site. The first thing is rather obvious. We have the ring closing reaction, makes things more rigid, which is good, which is fine. But we also get a little bit more of radiation here, because in this case, we have an additional interaction with an aspartic glutamic acid down there with an additional amine, or nitrogen. But you can also downsize the ring system here with a thiophene and also put the ring closure into motion. So this is something that retains at least the profile, which we've seen in the original compound, and also the intermolecular H-bond interaction. What I really found interesting is actually a series of compounds that was also proposed by Recall, namely compounds that also change the protonation state. So ultimately, this is quite nice because if you do stuff on with Recall within CSAR, all protonation states are calculated on the fly. In this case, you can actually see that if you would like also maybe to increase the solubility of a compound and introducing a secondary amine, you can also work with aliphatic systems or aliphatic ring systems. In those cases here, we also, I would say, a rather unattractive scaffold. Yeah, because let's be honest, this does not work out. Nevertheless, it actually, in this case, shows us quite nicely. We could actually introduce a spiral system here to really fix everything, to maintain the distance, and put also the nitrogen in the right coordination, and also introduce another additional H bond to the pollination state here. So my giveaway for this whole case study here is you can also use Recall for those kind of things, especially to really capture the whole idea behind a compound. And also by applying pharmacophore constraints, you really can guide your searches and come up with very decent solutions. Just a side comment. this whole WeCore index, as we call it, by Adel, namely the magic rings, is quite nice because it contains no decorations. It's only ring systems without any claws, without any metal groups, stuff like without any chlorine, without any metal groups. So basically, pure undecorated ring systems to especially work with those cases where you really would like to work with different scaffolds. Okay, what's the takeaway so far? Now for the first part, modifications of functional groups in regards to replacing them with a bioisostere can change geometry. And this is something you should absolutely keep in mind, which will be highlighted also in the next section. Next thing is that proximal interactions are way more stronger than distal interactions when it comes to intramolecular interactions, be it H bonds or be it sigma hole interactions slash chalcogen interactions. Always a nice thing to introduce if you would like to rigidify your compound and stabilize an active conformation. Bioisosteres can also be implemented more efficiently if you not look only into the functionality itself. So do not look at the amide, but look at the amide and the ring that is attached to that, or the subsequent one, doesn't matter. By doing so, you have way more degrees of freedom to work with and also way more ways to rigidify the compound, which can reduce rotatable bonds and therefore also improve bioavailability and other parameters. Last but not least, I highly recommend do things in 3D. It doesn't, even if you don't have a structure in front of you, so basically no target structure. Also, ligand-based drug discovery can work in 3D, where you just overlay your compounds. You can use Caesar similarity scanner for that to check, is my bioisosteric replacement a good idea or not? Because geometry changes, and if you would like to maintain an active conformation, geometry is important, and geometry only happens in 3D. Okay, this was the first part. Let me take a quick look at the questions if there's something urgent I need to disclose. Not so far. Okay, wonderful. Then, part two. One ring to rule them all. Again, I am a lazy person. I've taken a dive into a review to see what's the most prominent ring system. And to make a long story short, we now zoom in into the top 100 compounds and zoom in into the most ranked ones. And it's not even a competition, one order of magnitude higher. It's the phenol ring system. The phenol ring system is the most prominent structure of FDA-approved drugs, which will be also the highlight of the second part of this webinar workshop. The phenol ring system. Boring but beautiful. Just to browse through, we have 6 carbon atoms, we have 6 hydrogens, 6 120-degree angles, and 1 hexagon shape. This is a very aromatic and lipophilic group, and it is absolutely not eligible for any H-bond interactions, at least not any meaningful ones. Some people will now phone, but there are sigma hole interactions possible or stuff like that. Yeah, but let's be honest, in the context of drug discovery, there are very rare examples for that. Ultimately, the whole driving force about the phenol ring system is actually lipophilicity. What people do not always have in front of them, but I think it's more or less obvious, but it needs to be phased out. Phenol ring system can have different roles in drugs. One is they may form interactions with the target. Lipophilic interactions, aromatic interactions, it's there, they can form them. But sometimes phenol ring systems are just a linker. They are not contributing that much to the overall binding affinity, but they couple two different pharmacophores nicely without contributing that much. And if you really scroll through the PDBs and you pay close attention to phenol ring systems, it's quite prominent that those ring systems are just there. The binding site can be quite hydrophilic in some cases, which is not bad, right? The phenol ring system doesn't lose anything when it's inside a hydrophilic binding site. It does not decrease the affinity, but it does not increase the affinity. So therefore, it's the same as if it would be in water, it's just neutral, but without contributing to the overall binding affinity, it's just a very spacious linker. What people really forget sometimes a phenol ring system is just there, especially if you have a high throughput screening or virtual screening. Sometimes it's even solvent exposed. It's just not contributing anything at all. And therefore, if you have a hit compound, I always recommend to check, is the ring system really worth it, or could you just get rid of it? A nice hint from my side, if you want to check how well a student has prepared for an exam, ask him to draw a final ring system. The faster and the more sophisticated it looks like, the more the student has draws and structures. This is also what I have used when I was teaching students, but it's just a side story. Okay, what are the problems with phenols? Obviously, very lipophilic, so very insoluble, can be a huge problem for drugs. The second part is the aromatic oxidation. In some cases, or the pure benzene ring system, they are metabolized by the CYP enzymes, basically introducing oxygen, and subsequently with a cascade, they end up with those covalent groups that bind to the cysteines of the CYP enzymes, basically leading to covalently, basically leading to hepatotoxicity. Another thing we should always keep in mind when you talk about phenols is their low novelty. So basically, it's quite hard to have more stuff attached to them. It's nothing special by the end of the day. And they are quite planar, which also limits modification sites. Just to conclude this selection of the metabolism, there's always a good way to prevent metabolic stability. Always the best thing to do is introduce fluorines in the metabolic hotspots or electron withdrawing groups, but also steric hindrances with metal groups and the stuff mentioned here can be quite helpful to stop the metabolism of phenol ring systems. When we talk about phenol ring systems, there is absolutely one thing that we should absolutely keep in mind, namely the pi-pi interactions. This is something of personal interest for me because I am not a huge fan of pi-pi interactions, and even Grimmer proposed back in 2008 that we should avoid the term of pi-pi stacking, pi interactions for drugs ultimately, and just use pi-pi stacking interactions, but only on multi-ring aromatic systems. Two things, or three, there are even more, but ultimately those are the most prominent orientations that are observed, namely the T-shaped pi-pi interactions, the offset interactions, and the face-to-face interactions or the sandwich interactions. This is what people pay attention to when I watch them evaluating poses. But again, we should keep one thing in mind, namely, what do you think is the energetically most favored orientation of those three, among those three? I give you 1, 2, 3 seconds to think about that. And here is the solution. Ultimately, the T-shaped and the offset orientation is the most prominent one. And the face-to-face interactions where people say, oh yeah, this is exactly how things should look like, quite rare. This is offset, this is offset. But if you have two ring systems on top of each other, it's quite rare to be observed in crystal structures, and therefore please also pay attention to that when you evaluate poses during a visual screening campaign. Now, subjective word of truth, pi-pi interactions are overestimated. And why is this important? Well, people also pop up numbers. Yeah, pi-pi interactions can go up to, I don't know, 7 kilocalories per mole. Yes, but this is where Alverson really put everything into a nice perspective. More recent work, however, is revealing that direct electrostatic interactions between polarized atoms of substituents, as well as solvation-desolvation effects in strongly interacting solvents, must also be considered and even dominate in many circumstances. So what's hidden here? What's hidden here is interacting solvents, and especially strongly interacting solvents. When people talk about pi-pi interactions and they pop out those numbers are referring to gas phase measurements, calculations, or solvents like chloroform. I don't know about you guys, but the last time I have checked a biological system, there was rarely chloroform involved. It's always water, and water is a strongly interacting solvent that forms H bonds, right? So therefore, in the context of drug discovery, pi interactions have a very, 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 very low contribution to the overall binding affinity. Um, Again, there is just not enough data on biological systems that really supports pi-pi stacking as an energetically important interaction. Ultimately, you can boil down everything to solvation, desolvation, which is also the um, driving engine of the height scoring algorithm that we use. Just to accelerate things a little bit, I would now to talk about the phenyl bioisosteres. I didn't want to talk about functional groups in detail here. There's an amazing, one of the best reviews I have ever read in my life. And meanwhile, so if you would like to get a whole idea, please look at that. Just to highlight what the subsequent discussion will be about is basically the distance. If you talk about phenyl bioisosteres, you always have to think about, is it a terminal one, or do I want to connect two parts of the molecule and looking for replacement here? The distances are the most important ones. Anything else is up to discussion. So anything that is a phenyl biostere can be quite wide, which we will see on this slide. The variety is huge. We have sp2 hybridized systems of heteroatoms. We also have sp3 carbon-rich systems, or even sp3-rich hetero systems. There is a lot of things going on. Some of them are even charged by the love of God. You also can use, in some cases, linear functionalities, but this is something that is more directed towards terminal phenyl ring systems. All in all, the only thing that is of importance here is that you always need to check, does it match the binding site or not? So again, the return of the isotiazoles and the sigma hole interactions or chalcogen interactions. Two examples here, one compound on the left side, compound 16. Here they replaced the phenyl group for the isotiazole, and it was part of the whole recovery process just to stabilize the active conformation here. Just to explain the gap between over 1 micromole and picomolar binding affinity, they also introduced an macrocycle here to stabilize the whole conformation. But the highlight here is that the nitrogen and the sulfur form a chalcogen interaction in this case, which is even better than the phenyl ring system. And the next one, two studies published by Merck in 2021 and 2020 on the indolamine 2,3 dioxygenase 1. They had this compound and then subsequently came up with this compound, and they observed metabolic cleavage of this amide in rat serum, which led them to design of an improved metabolically stable derivative, namely here, which contains the bicyclopentane BCP as a phenyl bioisostere. In 3D, this is how it looks like. Quite nicely, they were able to get an additional interaction by introducing an additional amide here. As you can see nicely also here, we have a very similar distance between this carbon atom and this carbon atom. So this is really nicely, and they were also able to maintain bioactivity, which is quite nice. Similarly, or in the same spot, we can see this compound, daraplatib, a very, very promising candidate targeting lipoprotein-associated phospholipase A2 for the treatment of arteriosclerosis. It failed in 2014 in phase 3 after having a very promising phase 2. But this is how things are in the pharmaceutical industry, right? And what the GSK subsequently and the University of Strathclyde did in 2016, they published a paper actually considering how to improve the physicochemical properties of this compound, and they substituted this phenyl ring system again for BCP. Some drop in bioactivity here, almost 1 order of magnitude, but the physicochemical properties were improved quite nicely. What we see here in this example is an expert from the compound evolution series reported by Genentech on draw gamma receptor. This was the initial hit compound to some degree. We see nicely a tyrosine ring system here and then a phenyl ring system, which was also seeing here how they introduced a methyl phenyl group here, basically toluene, which affected coordination, right? So again, even if you have the same thing, but with one additional group substituent, it can change the geometry of the whole scenario. Please keep that in mind. And what they did quite nicely is this piperazine introduction. So basically, they made also an amide, basically mimicking this particular group here, and also to get rid of the charge. And this one was well reserved, and we can also see a nice increase in the bioactivity for this compound series here. So even without going into aromatic ring systems, also working with sp3-rich groups, we can also substitute or replace phenyl ring systems quite nicely. The last 3D example I have in front of me is this one. In this series, they improved compound 181A by actually merging those two ring systems into one. But more importantly, this is exactly what I was talking about when I've mentioned a linker, or does the phenyl system actually do anything? Here we can see, at least according to our software, that the contributions of the heavy atoms of this phenyl ring system are quite low, basically not existent. And what they did here, they actually introduced this heterocycle here to actually form additional interactions, which decreased and improved the binding affinity quite a lot. It also improved the log D value, make it more hydrophilic. And by introducing additional H bond partners, it would also able to form additional interactions with water, basically forming a pseudo ring system and rigidifying the active conformation. So I'm running late, but I'm still almost on time. Just to highlight two studies from Enamine, because I really appreciate how they really push things on their side in regards to building blocks and bioisosteric replacement. This was published previously in 2020 in Nature, and very sp3-rich phenyl ring system bioisosteric replacement. And Enamine published this one, not published, it was a preprint published in 2023 on this piperidine bioisosteric replacement. And they also synthesized an rupatadine analog containing this new motif here, which actually increased the solubility and decreased the lipophilicity of this compound. What's more important, even, they also were able to increase the metabolic stability of the compound by a factor of 11. Unfortunately, and I have to admit it here, they did not report, at least not in my knowledge, how active this compound was. But it seems like at least this compound is quite interesting when it comes to new modalities in medchem, medicinal chemistry. Last but not least, since we're talking about phenyl ring systems, those factors, those biostick replacements, Cuban, BCP, again, quite lipophilic, also not always metabolically that stable. And what Enamine did is they actually took a look into the advantages and disadvantages of this bioisosteres and actually made a very similar approach to their previously mentioned one and published it in Nature, coming up with this 2-oxo bicyclooctane as a successor of BCP here. They also synthesized a 3D analog of imatinib, replacing this phenyl ring system here, which led to an 10% increase in solubility, not much, but still, but an 8-fold decrease in lipophilicity and also a little bit of increased metabolic stability for this compound here. We are reaching the conclusions. What's the takeaway after hearing everything about the phenyl ring system? What you should always ask yourself, is this a linker or is this a terminal group? Because this is heavily important when making decisions. A linker needs to maintain the geometry of the compound, the active conformation. Terminal things are a little bit more flexible. So this is where you can play around a bit more, but linker needs to maintain the geometry. The next question or the next thing is my replacement actually even needed? So can I use it for introducing new interactions, like in the previous case for the heterocycle? And can I also abuse a biostic replacement to actually improve affinity? Last but not least, in regards to the takeaway messages, pi-pi interactions are absolutely overestimated by medicinal chemists and computational chemists alike. And the driving factor for binding affinity is actually the solvation-desolvation. My closing remarks here would be thinner ring systems are boring but cheap. Yeah, so if you work with limited funds, work with thinner ring systems. And once you enter the whole optimization stage, this is where you can really invest money because because bioisosteres will always be a little bit more expensive. So therefore, and also you need sometimes to synthesize a lot of the building blocks them yourselves because the availability of them is not that high compared to well-known ring systems. Bioisosteres are the perfect gate opener to really address patterns and limits, borders, whatever you want to call it, of patterns. And if you are smart enough and do things in 3D quite nicely, you can actually get quite neat ideas how to replace a compound. And last but not least, what people should always keep in mind, bioisosteres are extremely nice because if you switch from a very flat world into the 3D shaped one, you get a lot of more possibilities to actually introduce new groups to increase the binding affinity of a compound.